we are now getting into my PhD work. This is my wheelhouse, guys. This is what I spent 5 years looking at very, very extensively. So, how does this happen, right? We know that we have to bring things into the actual inside of the cell, but what about if those things that we have to deliver outside of the cell? So, in endocytosis, right? Endo meaning to enter, right? We are bringing cargo into the cell. In exocytosis, because we said this is another way that we can deliver molecules to where they need to be inside the cell, including outside of the cell. This means to exit. I can spell. And again, we are allowing our cargo to leave the cell. But how does this process occur? What happens is you have up in the left-hand corner here, you have your cargo protein, right? You have the thing that you want to deliver to the outside of the cell. And again, this is going to come really into play when we think about things like neuronal transmission, right? When we have our little vesicles, right, that we said wait for our calcium signal to deliver those vesicles to the actual surface of the membrane and release neurotransmitters, right? And then we activate the next cell. That is exactly what we're going to go through, but now we're going to look a little bit deeper into how do we actually dock those vesicles to the membrane to be able to release their cargo. So we have went through CME, right? We have went through endocytosis. This is the example that we gave. Right? And now we're going to look at exocytosis. So let's zoom in and look a little bit deeper into how exocytosis actually occurs. So for exocytosis, the first thing you're going to need, right? You're going to need to have whatever you want to deliver to the outside of the cell. So in this case, right, we have our little vesicle with the cargo that we want to deliver, right? So our cargo protein. And inside of there, right, we have a couple of different components. Our vesicle has attached to it some different things that are going to kind of help to guide, right? Think about the big balloons in the Macy's Day Parade on Thanksgiving, right? What do you see? You see those guys kind of walking down the street, and we don't just let those big balloons fly away, right? We have to actually kind of guide those balloons through downtown New York, right, by holding on to them. Think of all of these things that we're going to look at and walk through is basically our guides to getting our vesicle to the actual docking site to release the protein to the outside, right? So, if we have our vesicle with our cargo protein, our vesicle has attached to it 2 major components. It has first and foremost, right, what is known as a V-snare. We're going to come back to our snare complexes in just 1 second, right? But basically all this term is a vesicle snare protein. Now, let's just kind of sidetrack what snare is. Snare stands for, I'm not going to give you the full length of the actual term, but all I want you to understand about what snare is, it is a docking complex. Right? It is a vesicle docking complex. So as you can see, we've got some red strings, we've got some green strings, we've got some blue strings. All of those different colors, all of those different tethers, if you will, right, are all part of the docking complex that we collectively call the snare complex. So, on the vesicle we have V snares, and we also have a Rab protein. Okay, so that's the first one in the upper left, right? Kind of free floating. Then what do we see? In the second step here, right, we have to tether. Just like we said that we have to keep our balloon, right, to an individual anchored on the ground so it doesn't fly away. Not that we want our vesicle to fly away per se, but we want to dock our vesicle appropriately, right? So we need to tether it to the membrane. And again, we have another version of a different protein known as the tethering protein. So as our tethering protein kind of, grabs it along, right? Grabs and holds onto that vesicle, right? The tethering protein does this by attaching to the Rab protein on the vesicle. From there, we see our little blue T snare. Here comes another major component in our snare complex, right? So the third thing, our T snare on the membrane binds with the V snare on the actual vesicle. Right? At its very basic format then, these things wind up. They literally twist together, almost like Twizzlers, right? They twist together. And as they start to twist, right, they start to bring that vesicle closer and closer and closer to the membrane. Excuse me, until they fuse together, right? So let's look at that a little bit deep. Let's look at that a little bit, kind of further down the rabbit hole. So again, we have our V snares that hang off the side of our vesicle, right? And as our little tethering protein starts to kind of keep these guys in the general location of where they need to be on the membrane, right? They come closer and closer and closer, and the V snares are kind of hanging up there, waiting to bind to the T snares, right? I'm sorry, V snares are waiting to bind to the T snares who are on the membrane. From there, right, these two individual complexes wind together. As they wind together, almost similar to what dynamine does, it starts to cause friction. It starts to actually push the vesicle into the membrane, right? This all goes back to the fluid mosaic model. Because phospholipids can shuffle constantly, right? Because the fluidity of your membrane is about that of oil, right? In terms of salad oil, right? There is a very low viscosity that allows for vesicles to easily fuse with the membrane, right? Push those phospholipids all laterally to each other and fuse more membrane into wherever we're docking into, right? So again, exocytosis, typically we refer to these things as, when we're trying to excrete something out of the cell, right? Now, this also can happen if the Golgi apparatus is trying to bud off vesicles, right, to deliver to other parts of the cell. It also needs to exocytose different types of vesicles to deliver those proteins, right? So again, if we look then, there are a total that we have moved through, right, all the way back to that kind of very beginning rough outline of the cell in the three major ways that we can transport proteins. We can transport them into the nuclear pore. We can transport them across membranes, right? and we can deliver them via secretory pathways, exocytosis and endocytosis, right? Now, sometimes the ER does not appropriately make the right protein. Maybe we don't want our protein to go out because it wasn't appropriately folded, it didn't get the correct signaling sequence, maybe it just even doesn't have all the appropriate amino acid sequences, right? So this protein is not meant to leave the ER to be chaperoned or to be sent or shuttled to other parts of the cell. So if that's the case, right, then this very kind of rough rudimentary schematic of this, if we have a misfolded protein versus, say, one that is properly folded, right, what we have embedded in the ER is a kind of backup default mechanism. Is we have these things called chaperone proteins. And just like you're at a dance, right, and your chaperones are meant to be there to kind of, hey kids, hey everybody, let's keep it appropriate here, right? Think of the chaperone protein as pretty much exactly that, right? It's trying to determine and assure, most importantly, that the correct sequenced proteins are able to leave out of the ER, right, and be packaged in Golgi and sent out to other parts of the cell. So a chaperone protein is one way that we can guarantee that only the correctly folded proteins are able to leave the ER and go to other parts of the cell. Now, we also have other mechanisms in play, right? All of these, then, each one of these individual types of pathways, right? They're all involved as sensors to ensure that the misfolded proteins do not officially leave the ER. So maybe even my chaperone protein didn't do the correct job. Maybe my chaperone protein didn't actually cut off that misfolded protein. Don't worry, says the cell. I've got a bunch of other backup mechanisms to ensure that those proteins that are misfolded do not leave the ER, right? So maybe we have an internal cascade of activation, right? Maybe we just have an overall receptor that changes shape and that shuts the pathway down. Maybe, again, we may have signaling molecules that bind and they release maybe they just shut down protein synthesis directly in their tracks, right? The overall goal, though, is activation of chaperone genes, right? These things that make sure that we don't allow for these proteins that are misfolded to leave the ER. Activation of chaperone genes plus other genes that may have been activated by these pathways, right, all help to increase the folding capability and the folding capacity of the ER. So think about it. You have your production line. You're kind of assembling your amino acids. You're putting them together. You're trying to string them together to make a polypeptide chain. Maybe one polypeptide chain has to fold with another polypeptide chain to make a quaternary structure of a big, large protein that's now going to get delivered somewhere else inside the cell, right? All of these different pathways ensure we are correctly folded, ensure that we are only sending out those proteins that are needed, right? Because we have to think about the fact that sometimes, too, the cell is constantly receiving input, not just from internally, but externally. So maybe as you're going along and at the sign factory, right? The sign factory tells you that you need to print out stop signs, stop signs, stop signs, stop signs, stop signs. And then all of a sudden it receives some external cue that says, I need you to now switch, only make open signs, right? So now we have to shut down the cascade and signaling pathway of stop signs and shift now to open stops, or open signs, open signs, open signs, open signs, right? All of these different pathways help the cell to just constantly dynamically shift in terms of what the cell appropriately needs to do, right? So, again, these are all just backup mechanisms that are a good thing that your cell has in place to make sure that only the necessary proteins, only the correctly made proteins are actually able to leave the ER. Now, again, we said that should we need to release things ultimately to signal cascades to other cells, right? Because cells they're not in an incubator. They communicate all the time between each other, right? We may eventually have to secrete something to the external space so that it can communicate with another neighboring cell, right? That can happen through a multitude of ways, right? So the first way is that we can have unregulated exocytosis in the sense that maybe we need to put newly synthesized lipids. We don't really need to constantly monitor that because of the fact that we're always going to need new lipids, right? The lipid bilayer is so important, is so critically important that it constantly needs to be maintained, right? All the time. So we could call that a version of unregulated exocytosis. That's kind of the default mode. The cell says, yeah, of course we need phospholipids. Keep making them. Keep making sure we have enough to keep the cell protected. Or, right, an extracellular molecule, maybe from a neighboring cell, right, maybe from an above cell, below cell, surrounding cell, go ahead and it delivers extracellular signaling molecules that activate neighboring cells, right? And that allows for a signal transduction pathway, which we're going to talk about in chapter 16, to be activated.